Okay, this is the Osmosis lab, and I'm just going to show you. So this is dialysis tubing. So it acts like a semi-permeable membrane. So I'm going to twist the top. And tie a knot in it. If I had clips, it would work better. This is old school. Okay, so I tie a good knot there. Okay, then I open up the other end. Sometimes it helps to have wet fingers. There we go. I'm going to put a funnel here. Okay. And I'm going to measure out for every sample. I'm going to have 15 milliliters in there. So I'm not going to show you how to do all of them. I'm just going to do one so you can see. So this is water, and I'm going to, okay. And sometimes they'll, sorry about that, something fell. So I'm going to tie the other end. I'm going to leave space in case it gets larger. Okay. I'm going to knot it, and then I'm going to roll it on a paper towel to dry it off. Okay, and this is water. And then I'm going to place it on the balance so you can see, and this weighs 13.5 grams. So this is water, and it weighs 13.5 grams initially. Okay, so I'm going to do this for all the other solutions, and I'll come back. Okay, so I put all of these in the dialysis tubing. So this is water or sodium chloride in the dialysis tubing. So this acts like the cell. So the sodium chloride is in the cell, and the dialysis tubing is the plasma membrane. And I'm going to place it in 100 milliliters of water for 30 minutes. And I'm going to do, and then this is water. So this is 15 milliliters of water, and it's going to go into sucrose solution. I'm sorry, this is, water into glucose solution for 30 minutes. And then this is sucrose inside the cell going into water. And then this is albumin albumin is the protein that you find in egg white, and it's going into water for 30 minutes. And then I have our control, which is 15 milliliters of water in water. So those are going to sit for 30 minutes, and then we're going to mask them again. Okay, it has been 30 minutes, so I am going to take these out and clean them off, dry them off, and remask them. So this is the sodium chloride in water. So I'm going to rinse off. Actually, it's just water, but I'm going to rinse this off, dry it off. And I'm going to mass them. So this is sodium chloride in water. Please record the number. It changes, so what you do is that last number, you just, I keep touching it. Also the fan. Turn the fan on. Okay. So that's sodium chloride in the cell, and it's placed in water. This is water in glucose. I'm cleaning it off, drying it off. Okay, so that's the mass of water that's in the cell, and it was placed in glucose solution for 30 minutes. So record that mass in the data table. Now I have sucrose. Rinsing it off, drying it, and massing it. Let me zero this. Okay. And that's the mass of the sucrose that was placed in water. Now we have the albumin in water. The model cell was albumin. Dry it off. And mass this. That's the mass. And now I have water in water. I'm just going to dry it off. Okay. So now what you need to do is open up the Google Doc that's on in Google Classroom. Complete the data table. So hopefully you made the prediction first, whether or not you think water is going to move into the cell or out of the cell. You need to subtract the final mass from the initial mass and record the change in mass. You also need to identify if the solution inside the in the dialysis tubing is hypertonic or hypotonic compared to the environmental solution. And then for the analysis, you need to explain what happens with each cell model using the terms osmosis, diffusion, and semi-permeable membrane.